Does everybody understand this diagram? Huh? Well, this diagram. I know, you believe all the knowledge you have so far? Is that crazy? Look at that.
I have a question on this.
Yes.
I was doing note cards and stuff on it, and what is the whole, like, I understand that the straight solid lettings are what stimulate, but like the conditioned reflex and the brief reflex, are those?
I'm not going to worry about the specifics of the reflex.
Okay.
What I do want you to get from this is the 3 different phases.
Yeah.
What are some of the things that are key in those phases? So like cephalic, for instance, sight, sound, and thought. You know, communication with the central nervous system. And then
The stomach secretes.
exactly what the results are. Who's going to cue in on stomach? And a lot of these guys, even the gastric phase, the stomach is the key, and then intestines in the intestinal phase.
Okay.
All right, so understand the different phases of digestion. Who's this guy? This little blue Pac-Man looking thing. And where do we find him or her? I guess it's a him because it's blue. I don't know. What kind of cell? It's a parietal cell. And where do we find Mr. Parietal Cell? And this is basically the physiology of the chemistry of what's going on where. Yeah, stomach. The gastric gland. So, no. It is complicated. And what's going to happen in the book, and they do a very nice job of it, when we discuss the physiology of things, especially when we discuss systems, the physiology has to involve the chemistry as well. Certain things I'm going to say, you need to know that. Certain things, it's like, okay, I'm not too concerned about it. So yeah, the specifics of this, no. But I want you to get an appreciation for why the cell does what it does. And one of the things I want to point out is some of us, because of our body chemistries, and sometimes it's a genetic factor, overstimulate these cells and cause them to produce too much acid. Anybody in this class have that? Yeah. What's that? What might that result in? No smoking. No, the smoking might result in this, but what might it result in? Yeah, and what's that? Yeah, it might result in something called gastroesophageal reflux disease. Sometimes the overproduction of acid can result in ulcers as well. So there's a whole class of drugs that are going to help slow down the chemistry of the parietal cell, and they're called proton pump inhibitors. Anybody? Well, I've been taking them since I was in high school. Yeah, that whole class of drugs. And that's what it does. It helps to slow down the overstimulation of the chemistry, the physiology of the parietal cell in the gastric gland. No, I use it once a day. That's a once-a-day medication. I hate it. I wish I never had to take it. That's the only drug I take, but unfortunately. Yeah, well, that's the downfall of those drugs, because they also slow down the absorption of calcium. So you have to be very careful. If you take a calcium supplement and you're on a proton pump inhibitor, you have to make sure you take the right calcium. Did you know that? Yeah, calcium citrate. There's 2 products on the market, calcium carbonate and calcium citrate. You have to make sure you do the calcium citrate. Yeah. There's 2, the 2 I can think of off the top of my head is Caltrate and Citracal. Same company, but different component. So calcium carbonate versus calcium citrate. So somebody who has issues with too much acid production should take citric calcium citrate. So,
It's like a strong form?
no, it's just a different form. Yeah, and your body, because of its chemistry, is going to have an easier time absorbing citrical rather than caltrate, calcium carbonate, because of the chemistry involved here, because especially the carbonic anhydrase part of the chemical reaction.
Can you possibly, if it's possible, to explain this diagram just like simply?
First I told you you don't have to know this, and now you want me to explain it. It's basically very similar to what we saw when we looked at the red blood cell and the physiology of oxygen and carbon dioxide carrying capacity. So basically what's happening within the intestine, within the stomach, in the gastric gland, carbon dioxide and chlorine are two of the big players in this chemical reaction. So carbon dioxide and water, we've seen this reaction before, right? Carbonic anhydrase, that's going to result in dissociation and production of acid. Potassium is also involved, and the potassium channels are also involved in some of the chlorine shift, hydrochloric acid formation from parietal cells in the intestine. So this chemical reaction is going to lead to the production of hydrochloric acid. When I increase this chemical reaction, I increase the formation of hydrochloric acid. So with the help of these players, parietal cells secrete hydrochloric acid when we have increased carbonic acid formation, so that digestion in the stomach, the gastric gland, can take place. So this is basically showing you the chemical reaction of the production of hydrochloric acid by the parietal cells. And that secretion is going to go into the stomach. This is part of gastric juices. When I break things down, that acid environment helps also to dissociate hydrochloric acid. And the chlorine ions are going to help with this part of the reaction. They're going to help here with the production of the buffer system that helps to buffer acids in the system as well. And that's bicarbonate ions, HCO3.
In the book it says that arteries that supply stomach is more acidic than veins that go from stomach. So
You mean,
more acidic. I will show you. I say it right. Let's talk about the blood in the system that
right, but look at what's in blood capillaries around the gastric glands. It's more of the buffer. Bicarbonate ions are there to help neutralize the acid. So it might come in more acid and leave less acid. Is that what you're?
I think so. I cannot. Maybe later I will talk to you later.
Yeah. But that's what we see more of coming into the capillary beds at that point is the bicarbonate ions. Again, those are big parts of our buffer system in our circulatory system to help equalize concentration of hydrogen ions in solution.
Okay, I found it. Blood drained from the stomach is more alkaline than blood serving.
Correct. Because
That's
what?
why, yeah,
Correct.
Now I see.
Makes sense?
It's like a buffer in the system, in the cardiovascular.
Right, and it's going to pick up those extra hydrogen ions and help to increase pH.
Okay.
Yes? Good.
Thank you.
All right, so peristaltic waves in the stomach are going to help a couple of things. A, mechanical digestion. B, mix up those gastric juices. Because at this point, we want to make sure that we get that acid covering as much of the food in our stomach as possible. And then pH is going to reach a certain point, and that pH point is also going to help open up the sphincter muscle and get things moving into the next part of the digestive process. So what's the next part of the digestive process? Stomach emptying and putting food where? We're going to start the small intestine's role in the digestive process. So that's what retropulsion talks about. So we reach that certain pH point, and the pyloric end, and what's that sphincter muscle called? It's the pyloric sphincter muscle, is going to open up and allow chyme, which has now mixed with acid and sort of started to come down, come down in hydrogen ion concentration, but started to change in pH. We're going to open that sphincter muscle and start the process of forcing chyme. And what's the difference between chyme and a bolus?
Bolus that make the mouth. Before it will.
Chyme is what we call what?
Juice. Bolus mixed with the juice.
Exactly. The bolus mixed with all of the gastric juices. So that is going to then enter the small intestine and start the process further of breaking down. Now, not so much mechanical is going on. What do we have more of in the small intestine going on? Well, the nutrients are going in, but as far as digestion goes, because there's both going on in the small intestine, right? We're still going to break things down, and then as we move further along, we're going to eventually start absorbing. So chemical digestion is going to continue in the small intestine. So this diagram tells us a little bit about some of the different stimuli and inhibitory stimuli, that doesn't sound right, but things that are going to inhibit certain movements throughout the system within the digestive process. So presence of fatty, hypertonic, acidic chyme in the duodenum is going to do what? Once we hit the duodenum, do we want to keep squishing everything in the stomach and pushing it along? At some point, we have to stop that part of the reaction. That's the inhibitory part of the reaction. So that change in pH in the beginning of the small intestine is going to act to do what? Yes, decline what's going on in the previous part of the tube in the stomach. Slow down those contractions and allow the material to then enter the small intestine. So those are discussed under the regulation of gastric emptying, some of the different changes. Again, a couple of different receptors located in the central nervous system or communicating with the central nervous system, chemoreceptors and stretch receptors, are also going to have an effect on gastric motility through the central nervous system, sympathetic and parasympathetic divisions. Let me go back. So increased sympathetic activity, decreased parasympathetic activity is also going to take place to help with the whole rate of gastric motility. Yep. And in this case, basically think of, I know we think speed up, slow down, but basically what we're thinking of is pushing through or not pushing through. So we have to push through the stomach into the small intestine to start the process in the small intestine. Again, chemical changes are going to cue the next part of the tube. I keep saying that, right? But the next part of the tube needs cues not only for the tube itself, which is the small intestine, but a bunch of key players are also going to get involved here at the beginning of the small intestine. Those are the accessory organs to the digestive system. So, yes? From the chart that I told you, yep.
You said we just had to know the hormones, the site of production, and the
Correct.
target organ.
And that was table 23.1 on page 868. So you need to know those hormones, where they're made, and what their target is.
But we don't have to know what it does in that target, right?
You don't have to get crazy about the chemistry, but you should know that cholecystokine is important in activities that are going on with the stomach and the pancreas and the
Right.
gallbladder. Okay? Did that answer your question, Mila? Okay. So small intestine. These guys are going to come into play as well. And these guys, the guys I'm talking about in this diagram, are secretions that we're going to see from the liver, secretions from the pancreas. Now, we get a little help with secretions from the liver, from a little organ called the gallbladder. So when we enter the small intestine, we're going to see that there's different parts of the small intestine, and the cells, the lining, the mucosa, the inner part of the tube is going to change as well. We no longer have gastric pits. What we have is outward extensions of the mucosal layer, and those things are called villi. So instead of gastric pits, where we see the simple columnar epithelial cells come down like this, we see the simple columnar epithelial cells now lining the villi. So the mucosa changes. We're still going to see a lot of goblet cells here, and we're going to see that throughout the entire digestive process, lower digestive process. Because why do we need mucus? Why? We needed it, and we needed mucus for protection in the stomach, didn't we? That was going to help us prevent what? Yeah, digestion of our own cells. So mucus is protection there. In the small intestine, it's still protection, but mostly it's for propulsion, to help things move through. We have a much smaller surface area in this tube, not like the stomach, yes? So mucus cells or goblet cells in the layer are there to help propel food through the small intestine. Some of the changes in chemistry are going to initiate the secretions of those accessory organs to the digestive system. Again, a couple of things that are being secreted into the small intestine. One, bile. Who makes bile? No. The liver makes bile. Now, this is an analogy I use to help you understand bile. The liver makes bile. The gallbladder concentrates bile. So you know when you go to the supermarket and you buy clothes soap? Back in the old days when I bought clothes soap, the liquid clothes soap came in a big giant bottle, yes? And you'd have to incorporate several different helpers to get the bottle out of the cart and bring it home. And you'd have to put like 2 cups of detergent into the wash to make your clothes clean, right? Well, nowadays, the bottle has done what? That's concentrated. Same stuff, less water. So the liver makes bile, big closed soap bottle, and the gallbladder concentrates it. So when I add it into the digestive system during this process, I only have to add a little bit. I don't have to add tons and tons. It's also going to be more effective, just like the concentrated soap is more effective on our clothes. Lipid molecules, and this is a bad drawing, but hopefully I'll make you understand what bile does. Lipid molecules are hydrophobic. They repel water. They tend to hang out together in clumps. What happens, does anybody put olive oil in their water when they make their pasta? Yes. What happens to the oil? It floats on the top and it sucks together, yes? So this in your chyme, entering your small intestine. What do I have to do to each of these molecules in order to use them? I have to break them apart. So I have to take this, which is you know what I'm writing there? Yeah, that lipid molecule is made of glycerol. We're getting back to chapter chemistry. And fatty acids. In the digestive process, I have to break these bonds so that I can use these separate building blocks to make my own what? Make my own fat, make my own lipids. Maybe I have to take some of those fatty acids and make this. No bacon. That doesn't look familiar to you?
It's actually
There you go. A phospholipid. Or I have to make, it's all part of your plasma membrane. Or I have to make my own triglycerides that I use for storage in my body. So the food that I eat needs to come out of this ball. I need to take each separate molecule and I need to give some enzymes or apply some enzymes to break it down. So that ball is going to enter my small intestine. How am I going to break them down? How am I going to break that one down? I got to get them apart. So I have to somehow pull these apart so that I get them alone. That's what bile does. It doesn't break it down. It breaks it apart. It's called an emulsifier. That's what soap is, you know. Did you know that? Soap is an emulsifier. It's going to help break the hold of some of these lipid molecules on each other apart so that I can free them to then break it down. You're breaking apart some lipids, and sometimes some soaps will make you feel what? Really dry, because what do? They break down sometimes too much some of the lipids that you have on your skin that help keep you soft. So then you go and you buy some special cream, which is mostly made of what? No, lipids and water. Lots of water, exactly. And you add them back. So the secretions from the liver, concentrated by the gallbladder, are going to help emulsify fats. The other secretions we're going to see are coming from the pancreas. And the pancreas is a busy little organ. It's an endocrine gland and it's an exocrine gland. What the heck does that mean? Yeah, and what hormones? What did we talk about in the endocrine system?
insulin,
Glucagon. Okay, and that's the endocrine part. The exocrine part. Digestive system. We're going to make chemicals, enzymes, to help break down further molecules into their building blocks. So once I hit the small intestine, I'm talking building blocks. I'm going to take my lipids and turn them into glycerol and fatty acid. I'm going to take my carbohydrates, complex, and turn them into simple. I'm going to take my, what else? You had some for lunch. Chicken. Proteins, and turn them into what? Amino acids. Do you see where I'm going? So that's the goal here. So we have a whole slew of different chemicals secreted not only by the accessory organs of the digestive system, but also secreted by the cells that line the small intestine. Remember, I think I described this to you. If I look at a cell that's lining a villi, one little cell, those little cells also have microvilli. And these cells, if I put them under the microscope, kind of look like a brush. Little Bart Simpson. Got a little what's that called, that haircut? I can't think of it. Yeah, like a flat top. These cells collectively produce a bunch of enzymes. In that chart I told you needed to know, they're called brush border enzymes. So because they look like a brush head, it's further on down in the next part of the chapter. Is it that one there? Yeah, it's that one there.
you mean enzymes
Yep. So the brush border, the cells that line the small intestine, produce brush border enzymes. So we have secretions from the liver, we have secretions from, well, the gallbladder, secretions from the pancreas, and secretions from the small intestine. All of those are what composes intestinal juices. And they start the discussion of that on page 877. So, if we look at the cells in this diagram, which is on page 877, we see the microvilli, those little extensions on the cells. What all of these villi and microvilli do is create much more surface area. The more surface area I have, the more work I can do in a small space. So if I took the cells and the villi and flattened them out, how many feet of small intestines do I have?
About tennis field.
No, if 21 feet of small intestine, but if then I took the lining and stretched it out, I'm going to increase my surface area that much more.
It says in the book that it's
Tennis court? Yeah.
So does it like double, so it'd be like 30 something? I mean, you said 30 is like 60 something feet?
Well, if you flatten out the lining. So you're going to, I don't know what the diameter of a tennis court is, but it's at least 3 times that, right? 20 square meters. Yeah.
At the bottom of the page, right?
20 square meters. So, the liver and gallbladder work hand in hand. Can you digest lipids without a gallbladder? Yes, but same, think of the soap. Think of the detergent. Can I eat a Kentucky Fried Chicken anymore if I don't have a gallbladder? Probably wouldn't be very happy when I was finished because my less concentrated, exactly, it's not going to be as efficient at emulsifying those lipids. So a lot of those guys, rather than breaking down to this, I might break down to that when I eat a high-fat meal. Exactly, and they're just going to keep going through the digestive system and cause all kinds of problems near the end.
So what happens with the inflammation of gallbladder? Why?
Well, think about it. Have you ever made rock candy?
No, but I just know people that make it.
Well, how do you make it? Do you know?
No.
Sugar.
Yeah, you take a very highly concentrated sugar solution. And all you have to do with that highly concentrated sugar solution is disrupt it a small bit. And what do I mean by disrupt it a small bit? Yeah, I stick a piece of rope in it. You have to heat it to get it concentrated. But then if I stick a small rope in and disrupt it, I cause the concentrated sugar to come what? Out of solution. So if I tend to eat high-fat meals and tend to high demand my bile and keep producing lots and lots of bile at a high rate, I might cause crystallization of bile, and that's a gallstone. Now what's going to happen is if you look at the anatomy of the gallbladder and the pancreas, they kind of share a common exit. The common, well, they produce it and concentrate it at a fast rate, and that can cause agitation and formation of crystals, rocks, gallstones, just like making rock candy.
Is there any way to get gallstones out other than having
It depends on how big they are, depends on where they are. Yeah, unless they're causing you problems and they enter that common exit, which we're going to look at in a minute, they usually leave them be. But they can cause an awful lot of problems.
There is different like chiropractic offices you can go to though, and they give you ways of how to cleanse your gallbladder, but not
Yeah.
this much.
Yeah, no.
At home.
No. And again, it depends on the severity of the stone formation too. Sometimes people have a lot of what they call sand, same thing, sludge. Anything that's going to disrupt that exit can cause serious problems with serious digestive issues. One of the big holidays, the eating holidays, I call them. You all who've worked in the hospital know what I mean. If you're working on the eating holidays and you're in the emergency room, there's no rest for you. Because how many patients come in thinking they're having what? Thinking they're having a heart attack, and they're not having a heart attack. It's just Grandma's delicious desserts cause them to have a gallbladder attack. Plugging up that tube, causing a lot of pressure, causing a lot of pain. And referred pain comes to the chest because of where the gallbladder is.
Sharp. A lot of people think
Exactly. Yeah, and it is. It's very painful. It's nasty. Yes, all the same sort of symptoms of a heart attack. So yeah, the big holidays, Thanksgiving, Christmas, Easter. Busy.
Can you sometimes just get a sharp pain in this area too?
It could be, gosh, anything. Yeah, it could be. So gallbladder, if I look at the histology of the liver, I see the liver is very busy, very highly vascular. But another thing I see here is a lot of these green tubes that are transporting bile and pooling bile for the digestive process. Sending it to the gallbladder, and what's the gallbladder going to do to it? Concentrate it, and then is going to share an exit with this organ. And who's this? This is the pancreas. So we see some glandular-like material, very similar to what we discussed when we talked the endocrine system, except what does this exocrine function do with its secretions? And the secretions come from a group of cells called the acinar cells that hang out in little balls or groups. Where are they going to secrete their product? Yeah, through a what? Through some kind of duct. So this is the exocrine function of the pancreas.
So it says that the liver has a function of detoxification.
Yeah, so there's tons of macrophages in the liver that are going to get rid of things like old red blood cells or toxins in the blood. Spleen is going to do that as well. But the liver helps to neutralize a lot of the different chemistry that's going on in the body when we break cells down.
So when it will go, it's kind of like when it go to the organs and then from organs to the liver and back to heart?
Well, it depends. If you look at the tree, that's our circulatory system. The blood, much of your blood supply goes to some of these major organ systems. It's going to do that throughout the entire day. So the liver, for example, is very highly vascular. So by the end of the day, hopefully you've gotten at least 3 or 4 times your entire blood supply through the liver at any given time. So that any tox, it's not always, no.
But it will go like
Correct. Just like blood to the lungs. Blood's not always going to go to the lungs every single time. Sometimes some of it's going to bypass and go to the cardiac circulation. So not your whole volume is going to go to these major organs several times during any given day. More so than other organs. Because of the fact that there's so many blood vessels. Just like the lungs, the liver, exactly. So these are some of the chemicals that we're going to see associated with breakdown in the small intestine. Again, that chart, what page is that, Mila? Yeah.
Page 893.
So page 893, we're going to see a breakdown of all these chemicals in all of the different, 883? No, that, if you look on page 893.
The carbohydrate digestion.
Exactly.
The protein digestion.
Right. And when we start talking about some of the different molecules that we're breaking down, carbohydrates, proteins, fats, and nucleic acids, we'll see these guys listed there. So that chart, I need you to know.
This one?
Yep.
chart.
We need to know all the pancreatic enzymes.
Yep, and who they break down. Okay, and most of them you know already.
What do you mean?
Most of them we've talked about amylase, lipase. We've already talked about some of the things like cholecystokinin. They're already listed in the other chart that we discussed. Okay, and the enzymes. Remember, enzymes break down, not necessarily proteins, but they break large molecules into small, and they're typically named for what? What they're breaking down. So lactose, for example, is broken down by who? Lactase. So if you understand what all the organic molecules are, which of course you do because you studied that last semester and you know them all by heart, you'll know what the enzyme is.
How about like trypsin, chemotrypsin, carboxypeptidase?
Trypsin and pepsin are going to break down who? Pepsins we already talked about them. Trypsin, lipids. Huh? It's on there.
No, it's actually, well,
Yeah, it's on that one. It should be.
Protein.
Yeah, proteins and?
This one, trypsin, only protein.
Okay. And where?
Small intestine.
Yeah, and where's pepsin?
In the stomach.
Correct.
Yeah, but there's a lot. Trypsin,
Trypsin. They're all derivatives of. So no trypsin.
Just no trypsin.
Correct.
But there's like different.
Right. But if you see chemotrypsin, what are you going to think of? Trypsin. Yeah, exactly.
Yeah, there's a hard one is carboxypeptidase.
Yep. Carboxypeptidase. Whatever. All right, so this diagram then on page 884. These are some of the different mechanisms that are going to signal the release of some of these chemicals that are produced by the accessory organs of the digestive system. So when chyme enters the beginning of the small intestine, one of the things we have to do is neutralize it. Even though the pH has started to come up, and that's what triggered the opening of the pyloric sphincter muscle, we have to neutralize it more. So what we're going to see is some of the secretions from the accessory organs are also very alkaline to help neutralize chyme as it enters. Those enteroendocrine cells in the stomach lining are also going to release cholecystokinin or CCK, and secretin. These guys are going to enter the circulatory system, and they're going to help do what? Yeah, they're going to make contact once they get into the circulatory system with the pancreas, and the pancreas will then do what in response? Start releasing some of its pancreatic juices, along with a lot of what? Bicarbonate ions. Okay, who's going to get the signal during the cephalic phase, gastric phase? Vagal nerve stimulates gallbladder to contract weakly. So that's the gallbladder's signal to do what? If it starts to contract, what's it going to do? It's going to start squeezing out bile from the gallbladder. So these two are kind of going hand in hand. Secretions from the pancreas, secretions from the gallbladder are going to exit via the common bile duct. And that's another problem that can arise with gallstones. Plug up those exits. Anybody ever have pancreatitis? That can be something that happens because of problems with gallbladder, gallstones, sludge. They can back secretions up in the pancreas. Anybody who's ever had pancreatitis? One word to describe it. Very painful. 2 words to describe it. So it's very painful because of the pressure that builds up in those areas. So we want to keep things flowing. circulatory system and secretions within the digestive system are going to signal the next parts of the tube. So, overview of the function of the gastrointestinal organs. This is nice because what's it do? Yeah, I love tables. Breaks down the information, takes tons of information from many pages and makes it what? Make it small, because that's the goal. Make it small. Few words to make us understand what's going on. So this one is going to help you pull together what's going on in these different places. Intestinal motility, again, that's sort of a repeat of information from figure 23.28, but those signals that we discussed throughout the beginning parts of the digestive process are going to help later portions of the tube secrete what they need to secrete and also get their motility going so that they can push through the contents from one place to another. Who's next in line? The large intestine. So when we go from small intestine to large intestine, what should we have done mostly? It's not all done, but most of it is done. We've broken down most of what we're going to break down, and we've absorbed most of what we're going to absorb. There's still some breakdown and absorption that happens in the large intestine, but most of it, the majority of it, should have occurred by then. Who controls stuff coming from the small intestine to the large intestine? Another sphincter muscle. The ileocecal sphincter, or valve. So, when food comes to that point, when what's left in the small intestine, I should say, comes to that point and triggers the ileocecal valve, we release contents, or start to release contents from the small intestine into the large intestine. A substance called gastrin is going to help with the motility of things moving through the small intestine and getting them into the large intestine. And that we released way back when. Where did gastrin get released?
Gastrin is in the stomach, right?
Yeah. So remember, secretions from one part of the tube can signal the next part of the tube. Why would I start moving things through the large intestine when I have something in the stomach?
Because it's pulling the dust.
Yeah, the stuff from the small intestine has to end up there. So a lot of times after you eat, you have to what?
Poop.
You have to poop. Yes. That's how you train a dog. Yes? Who's got a dog? What do you do? After he eats, he poops.
That's how the old people are too.
Yeah, and that's how when you train children. Exactly. Yeah. No, not IBS. But that's typically when gastric motility increases after you've eaten. So if you're going to train a child how to use the potty, and that's a sphincter muscle they have control over. Potty, I don't know. Why do we call it a potty?
Because I call it potty.
Everyone, we all call it potties, but why do we call it a potty? Good question.
So weird.
Like some people,
A pot.
yeah, some people refer to it as the pot, and
Yeah.
for kids.
Because that's what it used to be, was a pot that you pooped in and then you went and emptied it before the invention of the toilet.
And it was probably just, made to sound kid-friendly.
Yeah. So anyways, that's why you have to empty when something enters the stomach, because of those signals that we're getting from earlier on in the tube. All right, large intestine is larger than the small intestine, shorter than the small intestine. How much large intestine will we have?
8, 7, 8.
Yeah, 7, 8, 10 feet. So less than small intestine. We still have some work to do here in the large intestine. And what we're going to see happening in the large intestine is we're going to collect all of the waste, whatever we couldn't break down and absorb from the small intestine, that's going to enter the large intestine. So we're preparing, to get rid of waste products. But we still have to do a couple of things in the large intestine. One, we have bacteria that hang out in the large intestine. It's good bacteria. It's referred to in many cases as your normal flora. And that bacteria is actually going to break down and release some very important substances that you are going to absorb from the large intestine. For example, there's some very important vitamins that are released by bacterial degradation that you then absorb. Vitamin K, there's some B vitamins that are also, and what else are you going to do with respect to absorption in the large intestine?
Some of the metabolic waste from bacteria.
Yeah, that's going to go. So that's already in the tube, but I might absorb some other things besides some electrolytes, some water.
5%.
Yeah, so I'm going to absorb water, which is going to help me absorb those vitamins and help me absorb those electrolytes from what's left in the large intestine as well. That's why when something happens to cause gastric motility to increase too fast and you empty too fast, give me the technical term for that, diarrhea, what's one of the problems? You get dehydrated and you might have electrolyte imbalances. Certain diseases, third world countries, cholera, all of those diseases that cause that gastric emptying to happen much too quickly, cause patients to succumb to the disease. Exactly. And that's one of the things that we have to be careful of when we have too much diarrhea or vomiting. Same thing. You lose too many electrolytes, too much fluid. Pedialyte.
Oh my gosh, my
Yeah.
son used to drink that like there was no tomorrow.
Yeah, it's better than Gatorade because Gatorade has electrolytes but also has too much sugar, and sometimes the sugar can actually irritate the stomach lining, so it can kind of worsen.
When we mess up with the flora.
Sometimes we might have,
These bacteria.
correct, we might have vitamin deficiencies, anemias. Okay, so that's if we're emptying too fast. We're not, think about bacteria and their growth rate. If I clear out way too many, I have very few. They're not going to be able to do their job. Antibiotics tend to kill off some of your normal flora. The big push for substances on the market called probiotics. They help to add back some of the important digestive bacteria. Lactobacillus is one of the groups of digestive bacteria in your lower gut. So what's one of the foods you can eat to help? Yogurt, and that's because that's how yogurt is made, with that particular group of bacteria.
Do you like Activia?
No, not at all.
You don't like
It's
it?
horrible.
I
It's
agree.
way too Activia, if you know what I mean.
Really?
So too much of a good thing is not so good either. Yes?
What about fiber one?
That's different. You're actually adding cellulose to the diet, so you're adding bulk. What's the best way to move things through your system in a natural way? Eat food. Food.
The right food, though.
Correct. There's soluble and insoluble fiber, and depending on what your issues are, you might have, yeah. But that's the best way to get your nutrients and your fiber is to eat them. Supplements, again, certain diseases, there's always exceptions to the rule, but for the normal person, well, with Crohn's disease, right, what happens to the linings? They become inflamed, they might become destroyed. So there's all kinds of problems that can result.
With Crohn's too, though, they can have blockages, which then they would have to have sections of their intestines removed.
Right, or sections of their intestine might become necrotic as
Right,
well.
So then they have to have it repaired. But also with Crohn's, do they not absorb things in the same way? 'Cause
Not as well, because think of again, if I decrease the surface area because I've damaged tissue, I decrease the length in which I can do this.
My mother-in-law has it, and her daughter also has it, and like with her, wherever like traveling, then she avoids eating anything fatty, like.
Right, or anything she knows that'll cause her symptoms
right.
to be
And
exacerbated, yeah.
My sister won't eat like red meats and certain things.
And everybody reacts differently, everybody's system is different, but there are certain things that someone with the disease knows, I can't have that, I have to stay away from that. Yeah,
Now, I guess the detox can cause varices, so
correct,
cancer can prone to the same thing
correct,
because of the possibly lasting from
yeah. So, large intestine, we're going to absorb water, we're going to absorb electrolytes, we're going to absorb vitamins and get rid of wastes. One of the problems with the large intestine and its design is, you see this little tendon that runs along the tube? It's called the tinea coli. Does anybody sew? Make curtains? We're going to teach you how to make curtains. When you want to make something pucker, make a little, what do you call that? I can't think of the little wavy, puckery. Think of your curtains with the little wavy pucker. You know where I'm going? All right, what do you do when you sew? You make a big stitch and you pull on it, and it causes the material to make these little puckers or coil. What do they call those things? Why can't I think of the name of that? Ruffles! Ruffles, is that right? I don't know. You know what I'm saying. That's kind of what the tinea coli does to that tube. It's tight and it pulls on it a little bit and causes these little pockets to form. Those little pockets are called hostra. Where are we? Where are they?
It's right there.
It's in there somewhere. But yeah, it's the little pockets. Okay. This is where concentrating your stools too much can be an issue. What happens when I concentrate my stools too much? Yeah. I have to strain. Yeah, well, hemorrhoids down here at the bottom. And that's this tissue here, the hemorrhoidal tissue. And this is the rectum and the anus. That's at the bottom. We have a couple of different parts. So in the small intestine, we have different parts of the small intestine. As we push through the tube, the beginning is the duodenum. And then what's next? The ileum and the jejunum.
Jejunum is actually
The yeah, the
ileum, the last part.
correct. So duodenum, jejunum, ileum. Cecum, ascending, transverse, descending, colon. And then the end is sigmoid colon, which leads into the rectum and then the anus. This is where we find the sphincter muscle, the check valve to control things leaving the end of the tube. And that's the external anal sphincter, which is skeletal muscle. Which do we have control over that when we're born?
What do you mean?
Control over that sphincter muscle when we're born? No. How long does it take? The track is there, information from nervous system to sphincter muscle. But what do I have to do to that track before I have conscious control over that muscle? Don't train it. Myelinated. It's unmyelinated when you're born. And you have to create the myelin sheath around it. That takes a while because it's a pretty long track. Yes? About how long?
3 years.
About 3 years-ish. Yes? So we're not ready to have conscious control over that muscle until the myelination process has completed. Is it the same for every person? No. Some are gifted and talented. Some are a little slow at the myelination process. hate to say it, sorry to pick on guys, but men tend to be a little bit slower in the process than females. Yes, thank goodness my daughter was 2.5. My son, 3, 3 and a couple of months, yeah. So you have to have that myelination occur before you have conscious control over that muscle. Couple of terms.
I think women smarter than men because we created to be a helper for them.
No, I just think that we don't like, you think about our anatomy and diapers. Yeah, it's a little bit more uncomfortable for women to be sitting around in, yeah. So I think they're more aware of wet and messy diapers, so they tend to
Nowadays,
train early.
I think a lot of kids have a hard time because they make diapers.
I know, yes, exactly.
They absorb so much they don't feel wet.
They don't feel it, so they're not uncomfortable.
Let them go naked. We need to go.
No, then they can poop all over the house. Huh? No, just know that that's kind of my little bit of extra interesting anatomy information. All right, so some of the membranes that surround the abdominal pelvic organ part of the digestive system are what I refer to as our apron, called omentums. There's a lesser and a greater omentum, and they cover that abdominal pelvic region. One of the things we're going to find inside the omentums, besides it being a serous membrane, is little fat tabs. Now, what's the fat for?
Protection.
It's protecting the abdominal pelvic organs. But what else do I use fat for? And we saw them also on the large intestine. We have some energy, right? So those tubes and their peristaltic movement, we have little fat tabs sort of spread throughout the abdominal pelvic region to help provide energy. Unfortunately, women tend to have extra little fat tabs there for extra protection. For what? Babies. Yes? So those omentums, and we're not going to see these in the fetal pig. One of the reasons, that's one of the things we're not going to see when we dissect the fetal pig. They don't have the omentums. When we dissect something with omentums, they are nasty because they're greasy and fat and ugh. So we see them here in the diagram. Other groups of men right here on the top, we see the greater one here. And again, that's lifted up in that one. So we also have peritoneal membranes. We talked about membranes like this when we discussed the heart and the lungs. Visceral peritoneum, sitting right on the organs. Parietal peritoneum, covering the cavity. And what's in between? No, what's in between the peritoneal membranes? Fluid. What kind of fluid? Serous fluid called what? Peritoneal fluid. So what happens when we have peritonitis? Inflammation of the peritoneal membranes, overproduction maybe of some of those fluids, and that can cause what? Yeah, a lot of pressure. And that fluid, overproduction of peritoneal fluid, is called ascites. And sometimes when protein deficiencies occur, severe protein deficiencies, we can have leakage of fluid through the actual digestive system. So that's where I was going. So, you know, exactly, when you see the commercials on television of a severely malnutritioned child, you see the pot belly. That's not fat. What is that? It's all that water, all that swelling, all that fluid leaving their digestive system because of their severe protein deficiencies. Proteins are going to help balance fluids in the body. And when we hit the chapter on fluid and electrolytes, we'll see how important proteins are to help balance fluid. Some of the other membranes we're going to find here Besides the omentums and the peritoneum, we're going to find parts of the peritoneum that hold together the small intestine. Do I see it here? Yeah. That I want you to pay attention to when you dissect your fetal pig. Very highly vascular and lots of little lymph nodes. Why do you think there's lots of lymph nodes and highly vascular? Holding together the small intestine.
Because that's where a lot of the absorption is. Right.
Absorption into the circulatory system. And what else? Yeah, and the lymphatic system is helping with the fluid balance and also to make sure there's nothing nasty going on in the stuff as we absorb it into the circulatory system. So there's a lot of lymphatic vessels there. What else? Anybody read ahead? Why is there a lot of lymphatics in the small intestine?
Because we have to absorb fat, not absorb, but
Yeah. It's exactly what we have to do. So when we talk about absorption of lipids, these big giant molecules, they're a little bit too big to fit into the circulatory system. We're actually going to apply a coating to them that'll help them be more hydrophilic, and we're going to absorb them into the lymphatic system first. They're going to hitch a ride there and eventually get dumped back into the circulatory system that way. So we'll talk about that in a minute. So the mesentery is going to hold together the small intestine, and then we also have a very large group of membranes called the mesocolon, and that's going to help hold together the large intestine. When you go and dissect your fetal pig, is it going to look like this? Nope. That colon is actually all bunched together on one side or another. Okay, it doesn't look like the textbook. It's part of the membranes. I guess we could consider it part of the peritoneum, but they always refer to it separately. It's a peritoneal membrane. It's a serous membrane that produces fluids. But you don't typically see it referred to as peritoneum. You see it because of the distinction between large and small intestine. So, lots of tubes, lots of membranes, lots of fat. This end portion of the textbook mentions the bacterial flora. Remember, we're going to get some B vitamin absorption from that. It's going to metabolize a little bit, and also some important ions and fluid balance. When things don't digest as well, or there's extras left over, let me give an example. Who's a fan of the chili? Love the chili.
I have a hard time with it. I like
But one of the things that's a bad combination when we look at foods is when you have too much fiber and too much protein. Because somebody's got to slack off a little bit in the digestive process. Too much fiber, beans.
Oh,
Yeah. So more of those and they're good for you, love the beans, but if you're going to eat chili, you should probably eat meatless chili, yeah. Beans, you should have beans.
Yeah, actually, I
Yeah.
found that without meat, I can.
Yeah, it's a little bit easier on this phenomenon that I'm going to mention. When we supply yummy goodies to that bacterial flora, they are going to pig out on what's left over, all of those beans that we didn't quite absorb. And one of the processes of their digestive process, one of the waste products is gas, exactly. So they talk a little bit about flatulence and gas formation. And again, it's usually if you give them lots and lots to eat, they're going to eat and have one heck of a time and produce a lot of gas. Yeah. So that might happen.
So combination of a lot of fibers and protein, not good idea.
Too much, yeah. And then it's what your system is used to, too. If your body is used to working on certain substances, they break them down easier. Like, for example, have you ever not eaten vegetables for a long period of time and then all of a sudden decide that you're going to be healthy? You know where I'm going with that, right? So you go and you start eating the salads and the fruits and the vegetables and
What if you always eat? I always eat that stuff.
right, and your body's used to that. But if you're away from it for a while and then you come back to it, you might have some gastrointestinal issues associated with it. Work past that. Yeah.
Which vegetables don't have much fiber? Because as far as I know,
I can't think of anything.
it all has a fiber. So
Well, because fiber is cellulose. So if you're a plant, you have your cell wall and then your cell inside. So anything plant material. Some processed foods break the cells down more,
yeah, that's true.
so they're easier to digest.
I'm thinking about like if I want to eat chicken, let's say it's a protein, it's mostly protein. What is the best way to eat it with? If it's not
No,
like
it's
rice
best.
has a lot of
Well, and again, this is my opinion, and there's a whole bunch of there's tons of different research on what we call food combining. But some say, those who are food combiners, say that you shouldn't have complex carbohydrates with proteins.
right.
So, but vegetables with proteins, yes,
It's good or not?
it's good.
Okay.
But like starches or potatoes, those should be eaten. Exactly. And what's the best, steak and potatoes, right? So again, there's tons of research, there's tons of opinions, but you just have to be careful and eat what
It will be good.
works. Exactly. So, the defecation reflex, again, is a signal we get via the spinal cord, skeletal muscles associated with the rectum and the anus, and internal and external sphincter muscles. All right, here's the fun part. Physiology of digestion and absorption. Know this diagram.
You want us to know the diagram?
Yes, ma'am.
How about path of absorption? Because it's very complicated.
Well, path of absorption, who's going to absorb carbohydrates? Most of it gets done where? I don't want you to know the specifics on path of absorption. Where is it going to work? Let's go to there. Okay? So, when we talk about carbohydrates, proteins, fats, nucleic acids, these are the four classes of what? What are all those things that I just said? Begins with an O. Organic molecules, yes. So when we talk about those four, we're talking about organic molecules, large carbon-based molecules. I need their building blocks to make my own large carbon-based molecules, to make my own proteins, which, as you know from last semester, are chains of amino acids in a certain order based on what? Who's going to determine what my proteins are made of? Yeah, your genetic material. So when you eat things, what you're doing is making big things small so you can take those building blocks and then make the molecules you need for your body. So this part is the physiology of digestion. What are we doing? Why are we doing it? And where are we doing it? And this diagram's got it all. As far as the path of absorption goes, I'm not going to get crazy about that. What you need to know is what the food is, what the enzyme source is, and the site of action. So the first three columns, basically. So carbohydrates consists of simple sugars. So when I take carbohydrates in, they're usually in the form of disaccharides or polysaccharides. Remember this? Chemistry? So what do I have to do? Chemically, I have to break them down into monosaccharides. So starches are polysaccharides, oligosaccharides, and disaccharides, big molecules of carbohydrates. Some of the different enzymes involved in breaking them down into their simple are ones that break down typically the disaccharides, like lactose and maltose and sucrose.
But
We
the
want to...
actual enzymes are the lactase, maltose,
Correct.
sucrates, right?
Correct.
So
So what's lactose? Do you remember this from the chemical chapter?
sugar and
Yeah, but it's a disaccharide. It's made of glucose and fructose.
No. I mean,
Oh, sucrose is glucose and fructose. That's right. Lactose is galactose and glucose. So our goal with the enzyme is to make what?
simple sugar.
Simple sugars, monosaccharides. So galactose is a monosaccharide. Glucose is a monosaccharide. Fructose is a monosaccharide. Okay? There's also ribose, deoxyribose. Those are all monosaccharides too. So know what the enzyme is that's going to break these down. Who makes the enzyme? Saliva. We're going to find some enzymes. Pancreas. We're going to make some enzymes. And you're going to see brush border in pretty much every category. Okay?
We're going to know, like, you said that we have to know, but just want to confirm. So about absorption, like via
Nope, 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 nope. So again, food stuff, what it is, who makes it, and the enzyme, so the enzyme source, and then where's it's doing its business, in the mouth, in the small intestine. You'll see most of these are the same. Okay, so carbohydrates, amylase, in the saliva and in the pancreas, they describe it here. Then there's a group of them secreted by the brush border. You see them there. The dextrinase, the glucoamylase, the lactase, maltase, sucrase, those are all secreted by the small intestine. Protein digestion. What are the two things that should jump out in your head? Protein. Pepsin and...
Peptides.
Yeah, well, peptides, right? That should help you remember that, right? So pepsin's stomach. In order for pepsin to work, what do we have to have? It's actually pepsinogen that's converted to pepsin by what? What's happened? Yeah, hydrochloric acid. So that digestion is going to begin in the stomach. It's going to continue with the help of pancreatic enzymes, trypsin, in the small intestine because of secretions from the pancreas into the small intestine. And then, of course, we have secretions from the brush border that are also going to aminopeptidase, carboxypeptidase. See, you just keep seeing that pep, pep, pep.
Both of them, brush border and pancreatic enzymes, they make the same enzyme.
They're similar enzymes, yes.
So how we would remember?
Well, this is, and this is.
No. You see, it's the
Yeah, one of them is the same. Yeah. I probably wouldn't ask you a question that would be cruel. But I might ask you, this is produced by who and who?
Brush border and pancreatic enzyme.
Right. So think pepsin, trypsin when we talk about proteins. Okay? And again, what do we want to break down? We want to break polypeptides into, or what's another name for a monopeptide? It wouldn't be a monopeptide. It doesn't have a peptide bond anymore. What is it called?
Peptide. Just a small peptide.
Oh, that still has a peptide bond. What's the single building block of a amino acids? Okay. The word peptide refers to the bond between two amino acids.
Yeah, if it's broken, it's
So you wouldn't call it a monopeptide. You know what I'm saying? Does that make sense? So that's proteins. Who's next? Fats are a little bit more complex. Because of the fact that lipids are hydrophobic, I have to create a carrier for them. And you can see the process of emulsification, breaking up fat from a big glob, and then using chemicals to coat, create packages so that these guys can be transported in a fluid environment. And that on page 895, figure 23.34, discusses the extra little steps we need to do when we digest fats. So we have to break them apart so we can break them down. And what are some of the enzymes that are going to break them down? Lipase. We have some in the saliva, not really very effective because it doesn't work in an acid environment. We have a little in the gastric, not really effective. Where's the big stuff going to happen with respect? Yeah, when we hit that small intestine. So when we get that emulsification action from bile, gallbladder and liver, and then lipase from the pancreas is going to do a great job on breaking down in the small intestine. And then what's the other nucleic acid?
There you go.
There you go. Nucleic acids. What do we need those for?
building blocks for our DNA and RNAs. Phosphate group, sugar group,
Yeah.
and nitrogen
Nitrogenous base.
base.
Yes? What is this? Oh, sugar. She spilt all of her no. They're building blocks of nucleic acid. Nucleotides. So this is what we need to get from the DNA that we eat. Can you think of anything that you eat that doesn't have DNA?
Doesn't have
Didn't have it originally, ever. No, vegetarian vegetables. They're plant cells. They have DNA, right?
they have
Not much. Water doesn't have it, yeah? Or some of the ions like salt. People say spices. No, they come from plants. They have DNA too, right? So we take in their genetic material when we eat them, and what do we have to do? Break it down into its building blocks. So basically we're remembering what we learned in chemistry. So we break down DNA and RNA, which are nucleic acids, into what? Those phosphate, sugar, and nitrogenous base combinations, nucleotides. So when we have to make new DNA, why would we need to make new DNA? To grow what? New cells, right? No babies. Never mind the babies. Your own cells, your epithelial cells. They undergo mitosis all the time, right? How do we make new epithelial cells? Mitosis, yes?
I didn't hear, so who doesn't have DNA?
Who doesn't? Oh, things like salt and water. Everybody pretty much has, yeah. Everything else you eat came from, at some point, even that, believe it or not, comes from what? Lots of chemicals.
What is it?
Pepsi. Fructose, high fructose corn syrup.
No, it has aspartame,
Aspartame.
which is going to kill me.
Yeah. So if it's not living, it's going to kill you. Yeah. Okay. All right. So where does this action take place?
It will make you, it will strengthen you, right?
Maybe I have like inside
No,
the
there's
arm.
Kentucky Fried Chicken and Pepsi. Oh, yeah, that's right.
So don't say that. That's true.
All right, so where do we do most of this work for the nucleic acids? Small intestine. So the pancreas is going to have some enzymes that we secrete that are going to help with the process, but the big player here is the brush border that are going to help in the small intestine. So this is where we'll see most of this work done. A lot of this work gets done just by the whole mechanical process and the chemical process, but the fine-tuning, the building block part happens with these enzymes. Here's the chemistry again. Again, don't get crazy. I won't worry. I'm not going to get too crazy about it. Just have an appreciation for the chemistry involved in the absorption of some of these chemicals. You appreciate it. Mil is very appreciative.
We haven't heard any essay questions from you yet. Yeah, I was going to say that.
Yeah.
I thought we did.
No.
No?
You get the email anyway.
Well, I know, but
Yeah, I'll give you the email. You want it early?
early one.
Yeah.
Early one.
When's the next test?
You know what? This is the question.
Not next week, the week
Maria,
after.
actually,
After what?
It's the week
After, oh yeah, after, yeah, okay. Yeah, I'll send it out.
but the
Remind me though, because I'm a space shot.
Can I ask you? The thing is, it's supposed to be not next, but after next Tuesday. It's supposed to be exam, but I don't think we're gonna spend
Right.
3, lectures for those.
Right.
Nutrition and
So we might be a little ahead of
But the
ourselves.
thing is, our lab exam is next Thursday, and it just will be unfair if you will do 2 exams on the same day.
We'll have to see how it goes. We'll have to see how it goes. It would, since I give you extra time for the other one, I might give you extra time. I'm pretty flexible.
But
I'll have to continue the material, but if I wait and give you the test, we'll do what we did with the lymphatic system. That's okay. It'll be okay. Don't stress, Mila. Don't stress.
Yeah, we'll be great. Feeling it.
I just don't like when we already passed material and we have exams.
I know.
And it's already
Yeah, but it's got to be one or the other. And urinary system, honestly, there's 2 wicked easy chapters. Wicked easy. Integumentary system and half of the urinary system. It's not really that difficult as far as anatomy goes. Those are the 2 easier of the chapters in anatomy.
Oh, you mean
When are we doing those?
there's a lot of
Even the anatomy part of it is not complex. Urinary system's pretty straightforward. All right, I got to shut up. So, I'm not going to make you know the details, but understand that there is specific chemistry involved, specific receptors involved in the absorption. This chemistry you need to know. So, emulsification, digestion, and absorption of fats. You need to know the little chemical packages. And we'll pick up on Thursday with page 895, figure 23.34. Deal?
Is next chapter nutrition and metabolism? Is it hard?
It's a pain, and no,
Thursday?
because one of the things, well, no, no, because